Good morning, students. Today we'll study immunoglobulin A. As you know, 5 different types of antibodies are produced by the body against the antigens. Immunoglobulin A is a very important antibody that plays critical role in mucosal immunity. IgA is also referred to as SIgA, that is secretory IgA. IgA forms 15% of total immunoglobulin present in the entire body. IgA has two subclasses, IgA1 and IgA2, and it can also exist in dimeric form called secretory IgA, that is SIgA. In the secretory form, IgA is the main immunoglobulin found in mucosal secretions like tears, saliva, colostrum, and secretions from gastrourinary tract, gastrointestinal tract, prostate, and respiratory epithelium. It is also found in small amount in the blood. The secretory component of SIgA protects the immunoglobulin from being degraded by proteolytic enzyme. Thus, SIgA can survive in harsh gastrointestinal tract environment and provide protection against microbes that multiply in the body secretions. SIgA can also inhibit inflammatory effects of other immunoglobulins. IgA is a poor activator of complement system and opsonizes very weakly. This is the structure of IgA. These are two dimers which are joined with the help of joining chain, and along with them is the presence of secretory protein. So as I've told you, IgA, it exists in two isotypes, IgA1 and IgA2. While IgA1 predominates in the serum, where IgA2 is present in higher concentration in other secretions than the serum. IgA2, the heavy and light chains are not linked with disulfide but with the non-covalent bond. In secretory lymphoid tissue, for example galt, the share of IgA2 production is larger than non-secretory lymphoid organs, just for example spleen, lymph nodes. Both IgA1 and A2 are found in the external secretions, where IgA2 is more in colostrum, maternal milk, tears and saliva, where IgA1 is more in blood. Polysaccharide antigen tend to induce more IgA2 than protein antigens. So this is the structure of IgA1 and IgA2 dimer. In secretory IgA, the form found in the secretion, polymers of 2,4-Ig monomers are linked to two additional chains, such as the molecular weight of IgA is 385,000 Dalton, that is 3,85,000 Daltons. So two additional chains they are. Number one is J chain, number two is the secretory protein. The J chain, the joining chain, which is a polypeptide of molecular mass 15 kilodalton, rich in cysteine and structurally completed different from other immunoglobulin chain. Right? This chain is formed in the IgA secreting cells. The oligomeric form of IgA in the external secretion also contains the secretory protein of molecular mass 70 kilodalton. This molecule originates from the poly-Ig receptor, which is responsible for the uptake and transcellular transport of oligomeric IgA across the epithelial cell and into secretions such as tears, saliva, sweat, and gut fluid. IgA activity. The high prevalence of IgA in the mucosal area is a result of cooperation between the plasma cell that produce polymeric IgA and mucosal epithelium that expresses an immunoglobulin receptor called polymeric Ig receptor PIGR. PIGA is released from nearby activated plasma cell and binds to PIGR. This results in transportation of IgA across the mucosal epithelial cells and its cleavage from PIGR for release into external secretion. It means that the plasma cell will secrete IgA. IgA will bind with the PIGA R, that is receptor cells. The receptor cells, they will take the Ig in the epithelial cell through endocytosis, where the secretory protein is attached, and through proteolysis the receptor is released and SIGA is released into the mucosal surface. In the blood, IgA interacts with the Fc receptor called FcARI or CD89, which is expressed on the immune effector cells to initiate the inflammatory reaction. Ligation of FcARI by IgA containing immune complexes causes antibody-dependent cell-mediated cytotoxicity, degranulation of eosinophils and basophils, phagocytosis by monocytes, macrophages and neutrophils, and triggering of the respiratory burst activity by polymorphonuclear leukocyte. So in this diagram you can see that at position number 1, the dimer IgA is present. This dimer IgA binds with the receptor. It enters into the cell, epithelial cell, through endocytosis, and then the cleavage is there. SIGA is released out into the mucosal surface, which will then interact with the bacteria and neutralize or kills them. Sometimes what happens, the bacteria directly enters into the cell, maybe because of cut. That bacteria, it binds with the dimer IgA present inside the lamina propria. Then what happens, the antigen which binds with the dimer IgA enters through the receptor cell into the epithelial cells through endocytosis, and then the complex is released outside and neutralized. Sometimes the bacteria, it binds with the endocytosed IgA in the epithelial cells, then it causes the inflammatory reactions. Now, the transport. The polymeric IgA, mainly the secretory dimer, is produced by plasma cell in the lamina propria adjacent to the mucosal surface. It binds to the polymeric immunoglobulin receptor on the basolateral surface of epithelial cell and is taken up into the cell via endocytosis. The receptor IgA complex passes through the cellular compartment before being secreted on the luminar surface of epithelial cells still attached to the receptor. Then proteolysis of the receptor occurs and the dimeric IgA molecule along with the portion of receptor known as secretory component are free to diffuse out through the lumen. In the gut, it can bind to the mucosal layer on the top of the epithelial cell to form a barrier capable of neutralizing threats before they reach the cells. So IgA, it contains two important components. One is the J chain, second is the secretory component. It is a dimer. So we can say that IgA, it when changes to SIgA complex, it will not be affected by the proteolytic enzyme, and it provides protection against bacteria present in the secretions of tears, saliva, nasal secretions, colostrum, and it is present in the lumen as well as the blood circulation. Now coming to the pathology part, if the decreased or absence of IgA is there, it causes significant problems, that is in Neisseria gonorrhea, which causes gonorrhea, then streptococcus pneumonia, then Haemophilus influenza type B, all releases a protein which destroys IgA. So if these three important problems they are there, that is these pathogens they are there, then the decreased activity of IgA is there, which is causing severe problems. I hope the concept is clear. Thank you.